Biosystems and Biodiversity candidate. The title of her presentation, Saving Biodiversity with Rare Plant Conservation Translocations. Her work is supervised by Jenny McCune. Emma Neigel is pursuing her PhD in Biosystems and Biodiversity under Dr. McCune. Originally from Athabasca, Alberta, she attended Mississippi State University on a track and field scholarship. She received there her BS and MS in horticulture. Emma has worked professionally as a conservation horticulturalist for the Atlanta Botanical Garden for the past 2.5 years, and is now back in Canada to explore her love for ecology and rare plant communities, working with the McCune Lab. So I give you Emma Neigel.
Okay, good to go. 1 million species are now faced with extinction on a global scale. To fight this grim fate, we need to protect species biodiversity. We hear a lot about this buzzword, biodiversity, but what is biodiversity? Simply put, it's the variety of life on our planet, the 3 to 10 million species that call this place home. One species takes up the most space, us. What is the result? Perhaps you might recognize this familiar satellite image. Fragments of forest consolidated within boxes of agricultural fields, except nature isn't supposed to be put into cookie cutter boxes. It's supposed to be wild and free-forming. Land fragmentation due to habitat loss, primarily from urbanization and agricultural land development, has pushed some species to the fringe of their existence. Unlike humans, plants can't just pack their bags and move to a place more suitable. How can we give plants a helping hand? Perhaps we need to look at conservation translocations as a potential recovery tool to relocate wild species back into suitable habitat. Translocation is the intentional movement of a species for conservation benefit and involves reintroduction or creating a new population. Canada has translocated bison, swift fox, and many other animal species, but hardly any of our endangered plant species. Why are we lacking in the green department? Because it's not as easy as throwing down some seed and walking away like Jack and the Beanstalk. Plants require specific conditions to thrive. Our goal is to establish rare plant conservation translocations in one of our biodiversity hotspots of Southern Ontario, where most of Canada's rare plants occur. Collecting seed from known populations and using computer models to target the best new homes, we plan to grow experimental populations on protected land. Testing how well the models can hone in on the right conditions for each species will give us a clue if it truly is a lack of suitable habitat preventing dispersal, or if these species just need a little help getting their seeds to the right places to really put down their roots. In-field experiments will enable us to test how competition from other plants and soil disturbance may also play a role. Consider a plant, the wood-dwelling drooping trillium that lives in the deciduous forests of Southern Ontario. There is good habitat in these forested parcels of land, yet the species only has 2 populations left. Why? Well, it turns out its seeds are dispersed by ants. Imagine a tiny ant trying to cross a cornfield to get to another lush forest patch. It's not going to happen. So maybe the reason this plant is so rare is that it can't spread to a new place because of the fragmentation we caused. Plants don't have real estate agents or Instagram accounts, so it's up to us to listen through learning and provide research to ensure our rare plant species recover and thrive. By studying limitations of dispersal and the habitat needs of these rare species, we can learn how to give plants an insurance policy and safeguard Canada's plant biodiversity well into the future. Thank you.
All right, thank you, Emma. Jiajie Liang, Master of Science, Neuroscience candidate. The title of the presentation, Mice at Home and a Prediction of Alzheimer's Disease. His work is supervised by Dr. Majid Moharjani and Howard Cheng. Jiajie Liang is a current master's student in neuroscience. He has presently undergraduate degrees in both physics and computer science, and he's researching automated behavior analysis of rodents and looking to utilize his diverse knowledge to aid neuroscience research. So if you are ready, sir, I give you control.
Hello. Hello. Yep. It sounds correct. All right. Let's first talk about Alzheimer's disease. In human, Alzheimer's disease gradually affects our memory, our language skill, our learning skill, and eventually our skill in performing basic day-to-day tasks. However, the early signs of it are often very subtle and vague. To study Alzheimer's disease, we have mouse strains that will develop the disease over time, and many experiments to study their behaviors. However, the current behavior experiments has some disadvantages. Firstly, this experiment only show a snapshot of the mice status. To see the progression of the disease, they need to be repeated multiple times. The other problem is us. These experiments require human interaction with the mice, which can affect the mice behavior, the exact thing we are trying to evaluate. Not to mention human evaluation itself tend to be difficult to standardize and reproduce. Now, my research instead look at the mice behaviors in their cage, and to do it with minimal human interference with the help of technology. I use automated home cage controlled by credit card-sized computer. These home cages have camera and sensors that record the mice behavior and monitor its environment 24/7. I will then look at the recorded data in these three directions as my slides illustrate. One is mice day-night cycle activity level. Mice follow a stable 24 hours day-night cycle, just like us, most of us. Now they are nocturnal animals, so they are more active at night. As Alzheimer's disease is known to affect sleep in human, I want to observe if the stability of this cycle is affected over time. The second is what we call excursion behavior. This is when the mice leave its nest, explore around, and then return to it. This is a behavior it's retained even in a cage environment. I'm looking at what the path they take are, where they stop, and for how long, and when they return. As disorientation is one of the symptoms in human patients, I want to see if there are observable changes to mice. Lastly is behavioral patterns. When does the mice eat, sleep, groom, or stand up and look around? I want to use automated process to see if there are specific patterns and if those patterns will be disrupted, just like how Alzheimer's disease can affect our daily routine. My goal is to find mice early subtle behavioral changes in home cage that can be used to predict Alzheimer's disease. I want to know when they happen and how they progress as the mice age. This experiment not only provide application in disease diagnosis and progress tracking, but also animal care, drug and treatment testing, and even human patient monitoring. And all of these start with these little mice in their home.
Thank you. Tyler Stewart is our next competitor, Master of Arts, Cultural, Social, and Political Thought. The title of the presentation, Why Do We Stand on Guard for Thee? Detuning the National Anthem. His work is supervised by Kimberly Mayer and Dana Cooley. Tyler Stewart lives and works on Siksikaitsitapi, the Blackfoot Territory, where he is pursuing a Master of Arts in the Cultural, Social, and Political Thought program at the U of L. His current research focuses on the role of sound within the ongoing structure of settler colonialism in Canada and how artists use sound as a form of resistance and refusal against imbalances of power. Outside of academia, he works across sectors from music to museums to magazines, fostering multidisciplinary collaboration to inspire new events and experiences that encourage participation. They are meaningful to the communities from which they originate. So, Tyler, if you are ready, it's all yours.
Close your eyes and open your ears. In a society dominated by sight rather than sound, I challenge you to hear the world around you, but more importantly, listen carefully, especially to those voices that have been silenced over the years due to unjust social structures. Then take action and use your voice to address these injustices in Canadian society. This is the approach I take to my thesis research, which asks the question, in what ways does sound serve to uphold the ongoing structure of settler colonialism in Canada? My research investigates one of the primary audio artifacts of Canadian history, our national anthem. While Canadians are taught to sing it from a very young age in schools, we aren't taught much about the song itself, how it was written in French for French Canadians, commissioned in 1880 as an assertion of Quebecois identity against the continuing rise of English culture. How the composer not only spent a decade performing as a blackface minstrel, but was also completely opposed to the creation of this country. And that these French lyrics were written by an ultra-conservative judge who often courted controversy for his deeply religious and anti-modern views. And that the original English lyrics were actually gender neutral to begin with, but changed before World War I to express true patriot love in all thy sons command. Then again in 1968, so that God might keep our land, an explicit religious reference that did not previously exist. While national anthems attempt to unite citizens of the nation through sound, my thesis unsettles the peacemaker myth of Canadian history, examining both the content and the context of the anthem's continuing evolution. Using a research creation methodology, I have developed a choose your own audio guide adventure where listeners are challenged to consider how they are positioned within this ongoing structure of settler colonialism and to address their individual and collective responsibilities as Canadians. We all need to hear these hard truths about Canadian history, but also go beyond hearing and listen clearly to these voices that have been silenced. However, listening is not enough. Learning about colonialism does not stop it from continuing. We all have a responsibility to take action. Whether you decide to sing along with the anthem, remain silent, or take a knee, it is important for all Canadians to address where they stand in relation to standing on guard for thee.
Thank you, Tyler. Competitor, Kelsey Racicot, Master of Science in Neuroscience. And the title of the presentation, Why Did the Chicken Cross the Road? This is work that is under the supervision of Andrew Awoniak. Kelsey completed her undergraduate degree in neuroscience at the University of Lethbridge in 2019. During her degree, she completed her undergraduate thesis in which she studied the effects of domestication on the brain. Kelsey is pursuing a master's, continuing her research on domestication. And in her free time, Kelsey likes to play with her cars and spend time outside, at least when it's not windy. Kelsey, it's your turn.
All right, can everyone hear me? Okay. Why did the chicken cross the road? I'm going to try and convince you in the next 3 minutes that we can learn a lot from chickens and that they're more than just a character in a classic joke. Imagine you're a wild chicken, like the one on the left in the slide, and you're in the jungle in Southeast Asia, looking for food and searching for shelter. All of a sudden, you get caught by humans and you're put into a chicken coop where your mate is now chosen for you. Your food is given to you for free. You only have a small area to explore, and the only other animals that you interact with now are humans and other chickens. This is what happened 8,000 years ago when humans began domesticating chickens for the first time. But what does domestication even mean? So domestication is the process of housing animals in captivity and breeding them over many generations, while breeding certain ones together to get particular traits in their offspring that make them more useful for humans. These useful traits can be many things, but a common one is friendliness towards humans. Many people have pets, and I think most would agree that your pet gets along well with most people, as well as your other pets. This is because we wanted tame animals to live in our houses. So when we breed tamer animals together, we get even tamer animals in the next generation. However, domestication is complicated. And while we're giving preference to these tame animals, some other characteristics begin to arise as well. Domesticated animals develop other characteristics in addition to tameness, like curly tails, floppy ears, and smaller bodies. Not only do they develop these physical changes, but the brains of domesticated animals are also typically smaller. This makes sense because it uses a lot of energy to maintain a brain, and if they're living in a protected environment where food and shelter are given to them, they have no use for a large brain, and they don't need to waste energy maintaining extra brain tissue if they don't have to. So this is where my thesis comes in. I'm going to study the brains of these domesticated chickens, but why chickens and not another domesticated animal? Well, there's something that I haven't told you yet. Chickens have actually enlarged a brain region during domestication, which is the opposite change that we have found in all other domesticated animals. This makes chickens very interesting to study as they're undergoing a change that we haven't seen before in any domesticated species. Chickens have enlarged a structure at the back of their brain named the cerebellum, which is thought to be responsible for several things, but mostly movement coordination. So how is their brain different than other domesticated animals and why? That is the question I'm trying to answer with my thesis. If there are changes in the cerebellum, are there also changes in other brain regions, like ones that are responsible for vision, hearing, and cognition? The results of my thesis will help us to understand if domestication can change these important parts of the brain that are crucial to survival in wild animals, and whether these brain regions are still as important in captivity as they are in the wild. If domestication can change the anatomy of brain regions that are responsible for the senses and cognition, we can learn a lot about the importance and the function of these brain regions in both captive and wild environments. So why did the chicken cross the road? I'll let you know when I figure out what their brain looks like.
All right, thank you. So moving on, our next speaker, Seth Campbell, Master of Science in Neuroscience. The title of the presentation, Hacking the Sleeping Brain, Can We Boost Motor Memory During Sleep? This is work supervised by Masami Tatsuno and Bruce McNaughton. Seth is a first-year Master of Science student in neuroscience. He studies memory and sleep in rats using electrophysiology and computational techniques. Seth's background is psychology with a computer science minor from the University of Calgary. He enjoys hobbies such as video games, fitness, and sleeping in when he can. Seth, your turn.
This university is full of talented people, including many musicians. But for me, despite taking over 5 years of lessons and countless hours of practice, my musical prime was still, well, quite bad at playing the piano. Without musical talent and a poor reading ability, I had to memorize songs to play them. But this task of learning all the hand movements, timing, and coordination is difficult though. But what if I could improve this learning process without more practice? Perhaps even while I slept. Well, I hope to propose a project that could address my struggle with the piano. Tasks such as playing the piano, throwing a ball, and riding a bike all use motor memory specifically, and learning any task involves strengthening our memory of it. So how can I improve this from a neuroscience perspective then? Well, outside of practice, sleep actually plays a critical role in our learning and memory. When we sleep, we know that our brains actually replay memories from that day and other experiences we have had. This replay is not necessarily a noticeable dream, but rather the brain's way of trying to strengthen the experience to become a long-term part of your memory. This replay of memories is simply known as reactivation. So how can we improve motor memory specifically? Perhaps improving the reactivation will help. And that is the focus of my project, a new method of improving reactivation of motor memories to boost learning performance. And this method combines two techniques: one, deep brain stimulation, and two, targeted memory reactivation. Deep brain stimulation uses electrodes inside the brain to stimulate specific regions. And don't worry, this doesn't hurt nor wake up rats or the patients. My lab has recently found a way for deep brain stimulation to actually improve memory reactivation. But technically, this could be any memory being reactivated. We want a specific motor memory though, like playing the piano and not throwing a ball, for example. That's where targeted memory reactivation comes in, doing exactly as it says. It helps the brain reactivate a certain memory using a clever trick. In the experiment, I will play a sound each time the rat tries to learn a motor task. Later on, I will play the sound again, but when they are asleep, and this will make the motor memory reactivate. Thus, synergistically, these two techniques together may improve memory reactivation for a specific motor memory. So let's say my experiment works out, fingers crossed. It would be cool to help a rat learn a task faster, but what about us? Whether a similar treatment works or not in humans, such insight may still be useful later for rehabilitation research. For example, stroke is one of the leading causes of motor disability in the world, So imagine helping stroke patients better regain their lost motor skills. But for the everyday uses, motor tasks are still part of our daily life, and understanding how to improve it is invaluable. So here's to hoping it can even help mediocre musicians like me. Thank you.
And thank you. Moving on. Fabian Rohden is a Master of Science student in biochemistry. The title of the presentation today, Preventing the Evolutionary Escape of Viruses. This is work that is supervised by Hans-Joachim Weiden. Fabian's from Germany, where he obtained his Bachelor of Science and Master of Science in Technical Biology. He's a graduate student in biochemistry in the research group of Professor Hans-Joachim Weiden. In his research, he's combining laboratory and computational methods to investigate the rational design of biomolecules. And as part of his research, Fabian is also looking into science policy for improving the governance of biotechnology. So, sir, the stage is yours.
Test 1, 2, 3. Perfect. All right, so with the current COVID-19 pandemic ongoing, I'm sure you've all heard by now that viruses can mutate, and maybe even that these mutations can make the viruses resistant against drug treatments. Antiviral drugs target the necessary components a virus needs for proliferation, such as a viral polymerase. The viral polymerase is a molecule that copies the genetic material of the virus, and its inactivation prevents the generation of new viruses. Unfortunately, the viral polymerase can escape this drug inactivation via mutation. Mutations are changes in the genetic material, which in turn can lead to changes in the structure of the viral polymerase. These structural changes can block the functioning of the drug, as is shown on the left with the purple polymerase. And the problem is that this process gets selected for by evolution because it helps the virus to survive drug treatments. This is known as evolutionary escape and is the same principle that leads to multi-resistant germs through the overuse of antibiotics. Luckily, evolutionary escape comes at a trade-off for the virus. So the mutations that confer the resistances usually also decrease the efficiency of the polymerase. And in my research, I want to investigate and exploit exactly this trade-off. I want to run computer simulations of the polymerase and afterwards conduct network analysis. Within the network analysis, the polymerase gets represented as a map of nodes, here shown as diamonds. And these diamonds are the elemental parts of the polymerase and are further clustered into communities, here indicated by having the same colors. These communities and the connections between them are like a blueprint for the polymerase showing how it works. With this blueprint in my hands, I can show how different drugs, upon binding to the polymerase, create a mechanical signal that gets transferred through the polymerase. Mutations enabling evolutionary escape block this signal, as is indicated by the magenta diamonds in the lower center. My hypothesis is that two or more of these mutations together can block the functioning of the polymerase, just because the combined structural changes are too much for the fine-tuned functioning of the polymerase. If that's the case, those mutations can't happen at the same time, and if the corresponding drugs are used at the same time, evolutionary escape cannot happen, just because either one of the drugs inactivates it or the combined mutations do the inactivation. In my research, I want to identify which drug combinations can achieve exactly this effect. And with this knowledge at hand, doctors know which drugs they need to combine to heal patients with viral infections. Thank you so much for your attention.
The supervisor for the work is Rob McDonald. Claudia completed an honors undergraduate degree in biomedical sciences from Brandon University in Manitoba. During her honors thesis, she investigated how microplastics consumption can affect molecular signaling pathways in the liver and the intestines of mice. Claudia is currently a neuroscience master's student under the supervision of Dr. Robert McDonald at the University of Lethbridge. Her thesis focuses on blocking different receptors and ion channels that are involved in the calcium influx in the hippocampus in order to better understand how learning and memory works. So, Claudia, if you're ready, here you go.
Okay, can everyone see me? Okay. So calcium is necessary for bone health. We've all heard that before millions of times, but you may not know that it also plays a very important role in learning and memory. If you take a look at the slide on the left, calcium will enter a nerve cell through different receptors, essentially just ions that open and close and let the calcium through. And once inside, it acts as a signal, letting the cell know that, hey, something important has just happened, you should pay attention. Now, if enough calcium makes it to the center of the cell, its nucleus, it's like its headquarters, then a really big signal is sent out through the entire cell and neighboring cells, making them aware of this big thing that had just happened. And that's how learning occurs in a nutshell. Now, if the learning is recalled at a later date, then that's what we consider a memory. Recent studies have shown that if you block one of these calcium receptors, that learning can still occur under certain conditions. Now, my theory is that the amount of calcium flow required for the learning depends on the specific type of learning that's occurring. So in my lab, we look at spatial learning and memory. And this is to say, learning information that helps an organism make a mental map of the world around them and how they move through it. And deep in the brain, we have a structure called the hippocampus, and it's crucial for spatial learning and memory. So I've identified 3 different receptors that mediate calcium flow while learning in the hippocampus, and I will block them using calcium receptor blockers to test if this is really what's going on. I will train rats on what is known as the Morris water task. And here you have a circular pool and this opaque water, and there's a platform hidden just under the water level and visual cues all around the pool for the rat to orient itself. And rats are pretty awesome natural swimmers, but the water is kept a bit chilly so that they're motivated to find a way out. And at first you place the rat in and it has no idea what it's doing. It just swims around like crazy. You can see the picture on the left. But after a few trials over a couple of days, it can learn the position of the platform and just swim straight to it. So for 4 days, I will train rats to a platform position. And then the next day, I will block the different calcium receptors in the hippocampus and then train them to a new platform position. 24 hours later, I'll remove the platforms from the pool and do what is known as a probe test, where essentially I just place the rat in and I ask the question, where are you going to swim to? If my hypothesis is correct, then blocking all calcium receptors should prevent the rat from learning that new platform location. And during that probe trial, this should just go back to the old one. However, blocking 2 out of 3 should show partial learning. Now, with this research, I hope to improve our understanding of what to me is the foundation that makes us human, our ability to learn and form memories. Thank you.
Thank you. Leila Armstrong, PhD candidate, Evolution and Behavior. The title, Backyard Wilderness, Investigating Urban Wildlife and the Nature-Cultural Divide. This is work supervised by Louise Barrett and Peter Henze. Leila Armstrong is a Lethbridge-based artist who is currently doing a PhD in Evolution and Behavior in the Barrett Henze lab. Leila has a Bachelor of Arts in Fine and Performing Arts from SFU and a Master of Arts in Media Studies from Concordia University in Montreal. She loves visiting natural history museums and enjoying their early to mid 20th century dioramas. Her interests also include watching pre-digital animation and reading suspense and horror stories. Apparently, the more chilling, the better. So with that, I give you Leila Armstrong.
When you see wildlife in your backyard, do you still think of it as part of nature? If so, do you see yourself as part of that same nature? If people see themselves as separate from nature, if they see themselves as actors that impact nature but are not actually part of it, what are the consequences for protecting the biodiversity with which we engage every day? Given that 81% of Canadians reside in urban centers, it is important to understand how city dwellers define and interact with urban wildlife. Through an investigation into the urban ecology of Lethbridge, my aim is to have people rethink their conception of nature as something that's external and removed. My goal is to increase awareness of urban ecologies as spaces in which humans and other species must live and thrive together. In my academic work, I use artwork as a way to communicate the data I'm collecting to a much broader audience. Backyard Wilderness is an ongoing project of which public art, as you see in my slide, and exhibitions are a vital part. I want to create opportunities for people to pause and rethink their relationship to urban wildlife. Here I draw on the work of theorist Alva Noe, who argues that art exhibits us to ourselves. For Noe, art gives us the chance to catch ourselves in the act of bringing the world into focus. I want to encourage people to embrace the everyday and to think of their yards and gardens as spaces in which wildness can not only exist, but also flourish. Here, to achieve this goal, I asked Lethbridgians to help me map the variety of species with which we share our city. I do this by placing trail cameras in people's front and backyards to capture images of animals such as skunks, raccoons, deer, coyote, and a variety of bird species. So far, I've placed cameras in 50 yards and interviewed all property owners and renters. I have also undertaken 2 online surveys about urban wildlife, revealing that the vast majority of Lethbridgians see wildlife within city centers as a positive thing. For example, although deer are the most frequently reported animal, only 1.2% of respondents considered them to be a problem. It is information from the surveys, coupled with the interviews, that I hope to convey to people in my upcoming exhibition at Casa Art Center in fall 2021. The title of the exhibition is Burrows and Bungalows. Specifically, I want to know and show whether or not people do conceive of themselves as part of nature or as separate from it, and the consequences of these positions. Thank you.
Apparently, I am most challenged in hitting the wrong button. So we move on to our next competitor, Jamie Petersson, a Master of Science student in Neuroscience. The title of the presentation, as you can see, Metabolites Reveal Recovery from Stroke, work that is supervised by Gerlinda Metz and Tony Montina. Jamie Petersson has a Bachelor of Science in Neuroscience and Psychology from the University of Lethbridge. Currently, I should say, she is in her first year pursuing a Master of Science in Neuroscience under the co-supervision of Dr. Gerlinda Metz and Tony Montina. Jamie started working on her current project during her undergraduate degree and decided to continue her work into her master's to complete the groundbreaking work that she had started. Using a clinical cohort, Jamie is investigating metabolomic biomarkers indicative of recovery from stroke. And I give you, if you are ready, Jamie, it's your turn.
Hi. You might know metabolism as that annoying bodily activity that's constantly pushing for weight gain that you've been trying so hard to work off. Well, I know metabolism differently. To me, metabolism can give insights into everything happening in the body, from what you ate for breakfast, to your stress levels, to cancer. Metabolism tells all. Personally, my research looks at stroke metabolism. Stroke occurs when the blood supply to an area of the brain is cut off, and brain cells in that area die from lack of oxygen and nutrients. After stroke, there are a lot of chemical changes happening in the brain and the body. Metabolites are at the bottom of this chemical cascade, so they can give an integrated view into all the chemical alterations happening in the body. Metabolites are tiny molecules that are products of metabolism, including sugars, fats, and hormones. So, where do you find these metabolites? In my case, I'm finding them in urine and blood. You can get them from almost anywhere, including tissue, saliva, and even hair. The current techniques used for diagnosing and monitoring stroke are costly, time-consuming, and have limited availability. So these stroke patients aren't getting treated quickly enough to minimize brain damage. With my research, I'm hoping to change that. I'm trying to discover if a technique that determines the metabolic composition of individuals can effectively diagnose and monitor stroke. This technique, called nuclear magnetic resonance spectroscopy, or NMR, is cheaper and faster than current diagnostic techniques. NMR allows me to track changes in metabolites, and hopefully I can use these metabolites to see how well patients are recovering. If it can, then I can offer stroke patients individualized treatments based on their personal needs. This would even further reduce brain damage by providing the most effective treatment as quickly as possible. So how am I going to do this? Stroke patients' urine and blood are taken at 2 separate time points, several hours after stroke or before treatment, and 6 months later, which is after treatment. Clinical assessments of function are taken at the same 2 time points. Following this, I will determine significantly altered metabolites and compare them to clinical measures. Ultimately, by looking at a stroke patient's metabolic profile, clinicians will be able to match individualized stroke patients to their optimal treatment program, hence minimizing brain damage.
All right, thank you. We move on to our next competitor, Maegan Trottier, Master of Science in Health Sciences. The title of the presentation, Childhood Trauma and Coping with COVID-19. This is work supervised by James Sanders and Cheryl Curry. Maegan is a second-year Master of Science candidate in the Faculty of Health Sciences, specializing in public health. With a bachelor's degree in psychology, neuroscience, and behavior, Maegan is interested in the epidemiology of psychiatric and addictive disorders. Her Master of Science thesis is focused on the impact of adverse childhood experiences in the development of multiple types of addictive behaviors, including the addiction to drugs, alcohol, gambling, and video game playing. In her free time, Maegan enjoys road trips, camping, and hiking in the mountains. And so, if you are ready, Maegan, here we go.
For the past year, we've all been faced with a collective trauma due to COVID-19. Over the course of the pandemic, each of us has attempted to cope with new stressors in a variety of ways, with some diving into tasks at work, picking up creative hobbies, using drugs or alcohol, or passing the time with activities like gambling, video game playing, or exercising. While most of these activities can be healthy in moderation, some are more likely than others to lead to negative outcomes. For instance, we know that exercise can lead to better mental health and decreased levels of stress over time, while other activities, such as excessive video game playing, can lead to increased levels of stress. But what is it that makes some of us choose healthier coping mechanisms during times of stress than others? I believe that may be due in part to adverse childhood experiences. So adverse childhood experiences, or ACEs, are a collection of traumatic events that occur prior to one's 18th birthday and include abuse, neglect, and household dysfunction, which involves events like parental divorce or a family member being incarcerated. Past research has shown significant associations between ACEs and addiction, such that people with higher ACEs are much more likely to experience addiction in their lifetimes. Over the past year, we've seen a similar association where heightened levels of traumatic stress due to COVID-19 correlate with higher likelihood of engaging in negative behaviors. So how do ACEs and COVID-19 traumatic stress interact with one another? Currently, it's unclear whether people with a history of ACEs may be more or less reactive to current traumatic events, and therefore more or less likely to engage in addictive behaviors. This is where my research comes in, as I aim to understand the relationship between ACEs and COVID-19 traumatic stress and the ways in which they interact to impact how we cope. To do this, I conducted a survey of 1,700 adults from Alberta and measured their ACEs, their level of traumatic stress due to COVID-19, and how their use of alcohol, drugs, gambling, and video game playing has changed since the onset of the pandemic. First, I'll address whether there's an association between COVID-19 traumatic stress and changes in addictive behavior during the pandemic. If there is a relationship between these variables, which I expect there to be, I will then assess the impact of ACEs on this relationship. In other words, my research will determine whether individuals with the same level of COVID-19 traumatic stress, but different levels of ACEs, will be more or less likely to use addictive behaviors to cope. Results from my study may inform how we help people manage their stress and addictive behaviors based on individual factors such as ACEs. By understanding how these individual factors affect people's coping, we may be able to target interventions and create a healthier society post-pandemic. Thank you.
Our next competitor, Carolyn Simonis, who's a Master of Science student in Biological Sciences. The title, as you can see, Palladium is not our pal, how catalytic converters may pose a risk to our freshwater friends. The work is supervised by Greg Pyle. Carolyn is in her second year of her Master of Science program in Biological Sciences. Her research takes place in the Pyle Laboratory for Aquatic Health, where she studies the effects of platinum group elements on chemosensory function and related behaviors in rainbow trout. Prior to starting her graduate program, she graduated from the University of Lethbridge with her Bachelor of Science in Biological Sciences back in the spring of 2019. So Carolyn, if you're ready to go, we will turn things over to you.
Awesome. Just a mic test really quick to hopefully everyone can hear me. Imagine this, you go outside, start your vehicle, but instead of that normal engine sound, you hear this loud, unnerving roar coming from underneath. I'm sorry to tell you, but chances are your catalytic converter has been stolen. Now, I'm sure many of you have seen in the news that catalytic converters are a new high-ticket item for theft. Well, why is that? These catalytic converters have a secret. Aside from completing their normal job, which is removing toxic pollutants from exiting the exhaust of a vehicle, they have precious metals within them. To be more exact, platinum, palladium, and rhodium, all members of the platinum group elements, make up the interior coating for these autocatalysts. Now, heading on over into roadside environments, people have been able to find these metals, which it makes sense if you think about it. Over time, these catalytic converters will start to degrade and erode, and so they will emit these platinum group elements into environments that they frequent. Now, these metals are not going to stay within the soil and dirt forever. Things like runoff have the potential to bring them into new environments, such as a freshwater one. Now, we know that freshwater can be teeming with life. And so what happens when we introduce these metals? Now, fish use their sense of smell to act on a multitude of behaviors, to forage for food, discover mates, escape from predators. Now, previous research has shown that certain metals are able to reduce the olfaction or the smell potential within rainbow trout. And if a fish's sense of smell is reduced, it can no longer react to various odorants within its environment. And if this is the case, we can potentially see an impact on the fitness of the animal, which results in the reduction of their efficacy for those aforementioned behaviors. Now, my project looks at exploring platinum group elements and to see if they act as other metals and have an effect on chemosensory function and related behaviors in rainbow trout. Now, currently within the environment, field researchers are finding more palladium than any other platinum group element. And so what happens when trout are exposed to palladium? In our lab, we use a neurophysiological technique called electroolfactography, where we are able to assess a fish's response to various odorants. My preliminary results show that when fish are exposed for 96 hours to palladium, we see a reduction in their ability to perceive various odorants, such as social cues, as well as smell or food cues. Now, if this is the case, these fish, I will mention, were exposed to 100 micrograms per liter of palladium, a concentration that is much lower than what we can see in the environment. Now, although I am hopeful as we start to usher in a greener age that we will not need catalytic converters in the future, this isn't going to happen automatically. And during this time, platinum group elements are going to continue to accumulate within our environment in unprecedented concentrations. Now, Canada does have guidelines to protect freshwater species for a variety of contaminants, but platinum group elements are not on this list. Thank you.
All right. Anne Jones is pursuing a PhD in evolution and behavior. The title of her presentation, Where the Action Is, Playground Behavior of Preschool-aged Children. Her work is supervised by Louise Barrett and Peter Henze. Anne is a PhD student in evolution and behavior with a special interest in human ethology, development, and the history of education. She is also a certified Montessori preschool teacher. So Anne, if you are ready, it's your stage.
All right, I'm ready.
Okay.
When thinking about children's adventurous behavior on playgrounds, maybe you, like many parents, educators, and playground designers, first think of safety. But have you thought about children's need for exploration and risk-taking? Today's playgrounds scarcely resemble their original playgrounds. Compare the rickety 1900s playground in my slide to the prototypical playground of today. Perhaps the early playgrounds did need a bit more regulation, but over the last few decades, playgrounds have become more and more standardized, and so-called helicopter parenting has become the norm. All this, even though serious injuries on playgrounds are rare. Children need a healthy amount of risk to learn how to navigate their environments and understand their own limits. So I'm asking, what are the costs of an ultra-safe playground where children's play is constantly monitored? To answer my question, I'm looking at the everyday behavior of children. Laboratory research suggests that the more ways people try a new motor task, like standing upright on a bouncing board, the faster they'll learn. But I want to know how variable children are in everyday motor behavior, because the conditions in the lab don't always reflect the conditions of everyday life. For example, laboratory studies on how babies learn to walk usually force them to walk down a straight path, unlike the windy path that babies actually take when learning how to walk. That's why I'm conducting my research under naturalistic conditions at playgrounds, where I use observational techniques developed by researchers who study animals in the wild to systematically record the motor behavior of children while also recording the physical and social conditions under which actions occur. By recording the different actions that children perform on a play structure, such as bouncing, rocking, and swinging, I can create a picture of how variably children behave when in the presence or absence of caregivers and other children. I want to know, do children use structures for their intended purpose, or do they find unconventional ways to play on the structure? In other words, do children always use slides for sliding, or do they use them for climbing, hanging, and perching as well? I expect to find that in the presence of other children who demonstrate new ways to use the structure, exploratory action will be bolstered, while caregiver presence will impede it. Creating a picture of children's everyday play will provide data-based evidence to parents, educators, and policymakers as they weigh the costs and benefits of playground safety.
All right.